Good afternoon, champions. So let me just wait for some time and see if it's get loaded. Okay. Checking. Okay. It's loaded. So there is someone who is saying, hello Ammu. Who is Ammu? Oh my god. So we'll just wait for some time and then we will start off this class. I see 6 people have joined. Let's wait for one more minute. Yes, 12. Oh my god. Good morning. Sorry. Good afternoon, dear children. I hope you all are doing great and your KCET preparation is going on really strong. Can you give me a quick hi, hello, something, so that I know that you all are human beings out there? No hello, no hi. Hi, yes, the first hi came. J.E. Sharda, hi. Narsimha, hi. Hello, lot of people. Yes, that's the spirit. So today, my dear children, in the Rank Booster series, I am planning to cover 2 chapters. That is molecular basis of inheritance as well as principles of inheritance and variation. We'll start off with principles and inheritance and variation, and then we will move on to the next chapter. Works for all? All of you happy? Are you all happy? Girish, Radha, Sneha, Sharda, all of you doing great. So good. Good afternoon, my dear children. We'll just wait for one more quick minute and then we'll start off. Yes, happy. Sharda is happy. Himanshu is happy. Yes, that is what I want. Everyone should be happily preparing for your examination. Great to see that. Hi, Vijaya. Okay, so shall we start then? Okay, give me a thumbs up if we can start. It's all according, and please share it with your friends. Okay, all the people who are preparing for same questions come on the day of examination. I hope so. Well, same question, there are less chances, but yes, the same concepts could be repeated. And I am pretty sure if you learn the concepts, you would be able to do the questions as well. So I see a lot of you giving the thumbs up. So probably we'll start off with the first question. All of you, read this. So which of the following combination of genes are found to be loosely linked in Morgan's experiment on linkage? You are asked about loosely linked. Okay, you have to find that. So genes for yellow body and white eye, genes for miniature wing and yellow body, genes for yellow body and red eye, genes for miniature wing and white eye. How do you answer this? You answer like this: 1A, B, C, D. Please put down the number like 1A. Vijay says, I like your classes. So thankful to you. Okay, thank you so much for saying that. That's so kind of you. Yeah. So someone says C. Is the answer C? For first, you are asked in the question, my dear children, which one among them is loosely linked? Now, if you are someone who do not know the answer for this, I'll request you to go to 67th page number of your NCERT. Okay, from down 6th line, if you start reading, you'll get your answer. So loosely linked genes means basically they are far away from each other. The genes which are there, they are far away. If one is here, the other one will be here. And if the genes are far away from each other, do you think they will show recombination, my dear children? Children, keep on guessing. I have not got the correct answer yet, which is why I'm asking you to guess. Okay, so if these genes are far away from each other, will they show high amount of recombination? Yes. Okay, the recombination means this part can go to another chromosome, this part can go to another chromosome, making new combinations very much easy. Okay, because they are very far from each other. Now you are asked about such loosely linked genes. No one has given me the correct answer yet. So again, I am telling you, if you do not know the answer for this question, go to 67th page number of your NCERT, 6th line from down, you will find the answer for this question. So if the amount of recombination is more, that is, it is showing 37% recombination, that means it is loosely linked. If it is showing 1.7 or 1.3% recombination, that means it is, let's say, tightly linked. There is less amount of recombination, means they are very close to each other. Yes, Yashu, SSLC Gaming, yes, great, my dear children, Yashu, it's D. The answer is D. Miniature wing and white eye, both are loosely linked genes. So miniature wing gene over here, white eye over here, which is why they are known as loosely linked. Please understand the concept. It will be really easy for you to remember it then. Will you ever forget it? Can we go ahead with the next question? Your answer is genes for miniature wing and white eye color. So in your NCERT it is written, for example, he found that genes white and yellow were tightly linked. Okay, white and yellow were tightly linked and showed only 1.3% recombination, while white and miniature showed 37.2% recombination. That means they are very far from each other. Great. I hope you understand that. Can we move ahead with the next question? Second question, all of you. Try this question. It's okay if you got the first question wrong, it happens. Now that is how I have tried putting questions from your NCERT directly. Try this question, you have to answer like this. 2ABCD. 2ABCD. The pedigree chart given below, okay, over here, represents what? Autosomal recessive trait like myotonic dystrophy, autosomal recessive trait like sickle cell anemia, autosomal dominant trait like sickle cell anemia, autosomal dominant trait like myotonic dystrophy. Come on, champions. Come on, come on, come on. C or Shetty says 2B. C or Shetty says 2B. Okay, that's great. Nija Gunaradhya says also B. Shilpa says B. Yashu says B. Chandan says C. Okay, Jay student says B. Great, 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 my dear champions. The answer is actually B. So over here, how do you identify it? First of all, you have to know to identify the pedigree chart. If in the pedigree chart, the parents are not affected, but the children are affected, that means it is skipping generation. Okay, that means it is showing an autosomal disorder. Sorry, it is showing a recessive disorder. Okay, recessive. It is showing a recessive disorder, so it cannot be dominant. So the option having dominant, I have to cancel them straight away. Okay, so these options are going to cancel away. Now, over here, if you see, there is a boy being affected, there's a girl being affected, then there's a boy being affected, so it is not sex linked. It is not what? Sex linked as well. It is going to be autosomal. It is going to be autosomal, which is why your answer should be autosomal recessive disorder. So we have two options like that, autosomal recessive trait like myotonic dystrophy and autosomal recessive trait like sickle cell anemia. My dear children, myotonic dystrophy is not autosomal recessive trait, it is a dominant disorder. So there goes the wrong option, and here we have the correct option. So whoever said B, you all are exactly correct. So always remember, if you are going through pedigree chart, if it is skipping generation, it is recessive disorder. Okay, and in the sexes also, it is like boy girl equally affected, then that is autosomal disorder. Okay, done? Are you understanding? If you are understanding, give me some hearts. Give me some hearts. If you think that I should be slowing down, you can tell me. I am here to teach you. Ma'am, voice is slow. Okay, I'll put it over here. Does that work better? Yes, great, great, great. So I hope you're understanding. Great, my champions. Awesome. Thank you. Nijaganara, the SSLC, and all the children who are giving hearts. Can we try third question? A colorblind daughter is born when? Very important question. Okay, very important question. A colorblind daughter is born when? Mother is a carrier, father is normal. Mother is a carrier, father is colorblind. Father is colorblind, mother is normal. Mother is colorblind, father is normal. So 3B, okay, someone says 3B. 3C, I say 3B. Rishal, Kirtana, Sir Shetty says B. Yashu says, ma'am, voice is low. Still it is low. Okay, let me try over here. C. Okay, can we try? So, we want colorblind daughter. For a daughter to be colorblind, my dear children, her both the genes should be affected. Okay, both the genes should be affected. That means one gene is going to come from the mother's end. So mother basically should be at least like this. At least I am saying. At least she should be a carrier. Now in the case of a father, father should be colorblind definitely. Then only you can get this from here and this from here. Right? So your correct answer should be what? Mother is a carrier, father is normal. That cannot happen. Father will be colorblind. Mother is carrier, yes. Father is colorblind, yes, my dear children. In that case, it can happen. Now, father is colorblind, mother is normal. If mother was normal, there was no affected gene over here. She will be just a carrier because you should be needing affected gene over here, right? So again, this is wrong. Mother is colorblind, father is normal. If mother is colorblind and the father, my dear children, if he is normal, where do you get the another X from? She will be just a carrier. So that is also wrong. So with that, what do we understand? Your answer will be B. Okay, all of you who got B, kudos to you. All of you who got your answers wrong, it's totally fine. This is how we learn. So for a colorblind daughter, the mother should be a carrier and the father should be colorblind. Always remember this. Okay? Yes, great, great, great. Moving ahead with the next question, that is, what is an offspring of two homozygous parents differing from one another by alleles at only one gene locus known as? Okay, didn't understand the question, I can make you understand this. So basically I am saying, what is an offspring of two homozygous parents? Two homozygous parents as in, for example, I have capital T, and I have small t, small t. This is homozygous for capital T, capital T. This is homozygous for small t, small t. Now, they are differing from one another only by alleles on the same gene locus, as in same gene location. Same gene location. For the genes on the same location, that means these genes are coding for just one character. These genes are going to code for one character. If I had gene like this also, and I'm talking like this and this, they are separate locus, my dear children. In that case, this gene is going to code for some other character. But since they are coding for the same character, that is why they are on the same gene locus. So in that case, what will I call this? Dihybrid, monohybrid, trihybrid backcross. Okay, what will be answer? So 4B, I see lots of people. Kirtana says B, Nijaganaradhya says B, Shri says B, Vinita says B, Marappa says B, Nijaganaradhya says B, and Chandan says B, Vinita says B, Sneha says D. Baba, the answer will be what? B, monohybrid. Mono means one. Basically, only one character is being chosen over here. That is why it is known as monohybrid. If it was two character, then probably they had multiple gene locus. But talking about one gene locus, then it is one character. I hope you understood this. Okay, so this will be monohybrid, not dihybrid. If there were two characters, it would have been dihybrid. Got it? If you got it, please give me a thumbs up. If you didn't understand it, please tell me here. I am here as a friend to make you understand things. So do not think that I'll judge you for anything. Okay? So please make sure you're asking me the question. Okay, Narasimha said, great. Okay. Thank you. Children, are we in the same pace? Okay, are we matching the pace? Yes, I see certain hearts flying around there. Okay, cool. Now, when a pure tall plant is crossed with a dwarf one, the ratio of pure tall and pure dwarf in F2 generation will be what? I am crossing what? Pure tall. That means capital T, homozygous. Okay, now I am crossing it with pure dwarf, small t. And I am asking you the ratio of pure tall and pure dwarf, that means the ratio of capital T and small t in F2 generation. Can you tell me F2 generation? What will it be? So 5B, yeah. Who says 5B? Children, F2 generation. This is how we make mistakes in KCET examination. F2 generation you are in. So basically in F1 generation you have capital T, small t. Listen to me carefully, okay? I am crossing this and this, I get capital T, small t, this and this, capital T, small t, this and this, capital T, small t, this and this, capital T, small t. All of them are capital T, small t. This is F1 generation. You are asked about F2 generation. For that, what do you have to do? You have to cross between them, self-crossing. So I am taking capital T, small t, capital T, small t. I get capital T, small t, capital T, small t. Now I have to cross them. This and this goes, you get capital T, this and this goes, you get capital T, small t, this and this, capital T, small t, this and this, small t. So how many homozygous? Pure tall and pure dwarf is there? One pure tall and one pure dwarf. So your answer will be what? Yes, your answer will be D. So let me see who all said D. SSLC Gaming, no. Shri, and then Yashu, Suvidha, hi. I see new people, Sneha, Yogita, Rishal, and then Srinivas, Chandrasekhar, and yes, oh my god, Prathna. So many people are saying the correct answer. Rishal again, Aryan, Narasimha, Manjunath, Prathna, Ellen. Hello. Okay, so okay, CR Seti, lots of people are saying, great going my champions. So good to see that. So your answer will be 1:1. Please make sure you're reading the question carefully. Do not do the same mistake on the day of the examination as well. Okay, so all the children who got your answers correct, kudos to you. All the children who got it wrong, it's not a problem. We are going to clear that up. Yeah? Umar Sheikh, hi my dear child. Yes, now the significance of a test cross. What is the use of a test cross? To test the heterozygosity of F1 progeny, to test the homozygosity of the recessive parent, to test the heterozygosity of the recessive parent, none of these. None of these. Why do we need a test cross? 6B. To test the homozygosity of a recessive parent. What is the answer? Okay, I have A, B, C, D, all the things, options over there. So basically, I just want to find, your answer will be A. Why will it be A? I'll tell you why. Because basically, I want to find out whether I have a tall plant. I cannot see what is inside the tall plant, what is the genetics of it. So I want to check whether it is like this, that is whether it is homozygous or it is heterozygous. But mainly I am trying to find whether it is heterozygous dominant or not. I am trying to find this. So your answer becomes to test the heterozygosity of F1 progeny. Even in the question, if the option was like to find the homozygosity of dominant homozygosity of F1 progeny, in that way also it would have been correct. Now you might be thinking, why ma'am, why is it necessary? For example, I am a person who want to produce tall plants. Okay, I am a person who wants to produce tall plant. So I thought I take this tall plant and basically I'll try to fuse it. Okay, I'll try to, let's say, combine them. But then I realized what happened, I took this tall. So capital T, small t, capital T, small t. At the end, my dear children, I will be getting dwarf plants also. I do not want that. I want plants like this. So what do? I do a test cross. There, what I have to do is that I have to take the parent plant. Suppose it is the parent plant, and I will be crossing it with the recessive parent. And in that case, what will I get? Suppose this and this goes, I get capital T, small t. This and this goes, I get capital T, small t. This and this, small t, small t. This and this, small t, small t. So since dwarf children are being produced, I get to understand that basically it is not homozygous, it is heterozygous. I should not be taking this plant. Instead, now when I cross with a homozygous parent, it will be like this: capital T, capital T, capital T, small t, capital T, capital T, capital T, small t. All of them are tall itself. I want this plant. This is why we do test cross. So just to understand the heterozygous, whether it is heterozygous or not, I am going to do this. I hope you understood this. Your answer becomes A. Okay, so all of you who got A, great going. I hope that is clear to you guys. So it is not necessary that it has to be an F1 progeny. Okay, please remember, it is not necessary that it has to be F1 progeny, which is why some might be confused. But if you have an option like this, go for the first option. Okay, Riaz, hi Riaz. Now, 7th question. Which of the following statement is correct? Each back cross is a test cross. Each test cross is a back cross. Crossing F1 with F2 is a test cross. Crossing F2 with F1 is a test cross. Now, since I taught you already about that, you have to tell me the answer for this question. Baswasraj, hi. Which of the following statement is correct? Each back cross is a test cross. Each test cross is a back cross. Back cross means basically you're going back and crossing it. Ma'am, you didn't understand. You didn't understand the question number 7, or basically the previous question. Narasimha, can you tell me? So, basically over here, in this question, back cross means you're going back and crossing with something. That is what you mean by back cross. So here, what is correct? That is the question. Okay. Kirtana says B, SKC 4/7, D. Okay, Chandrasekhar, B. Pradeep is here, hi. B, A, B, C, D, everything is there. My dear children, each back cross is a test cross? No. Anything, for example, I have capital T, small t over here. I am basically just trying to, this is F2 generation, this is F1 generation. If I am trying to fuse with this, it is not known as a test cross. This is wrong statement. Test cross is only when I am taking a parent, okay, I am taking a progeny. It can be like this, it can be like this, homozygous or heterozygous, and I am fusing it with a recessive parent. With what? A recessive parent. This is known as test cross. Now, each test cross is a back cross. That means I have to go back. Yes, because what I am doing in each test cross, I am taking the progeny and I am going back and fusing with a recessive parent. I am fusing with a recessive parent. So your answer becomes B, not A. Now crossing F1, F2, this is wrong. Crossing F2 with F1, no. This is the only answer that we have. Okay? Your answer is B. So just a second, I am answering a question. How to download the session PDF? My dear children, basically in the WhatsApp channel, if you go, there will be links given for the downloading of PDF. If there is no link, I will tell the team to upload it really soon. Okay? Example of a back cross, basically just like this. For example, I have a plant over here. I am fusing with the F1 progeny over there. That will be a back cross. Okay, I am not worried whether this is recessive or not. But in the case of test cross, I am making sure I am crossing with a recessive parent plant. That is a test cross. Okay? Hi Kiki. Hello. So all my children, All of you who said the correct answer, kudos to you, and please go ahead with the question number 8. Until then, I'll be reading out the names of the people who got things correct. Yes, it is very much important to tell your names. Kirtana, Chandrasekhar, Jai, Pradeep, RX fans, Umar Sheikh, Angel, CR Shetty, Angel Gaming, and then Prathana, Sneha. Can you just check Bacha once? Suvidha, Bashir, oh my god, Anusha, a lot of people out there. Now we have the next question. Person suffering from Down syndrome will not show. It's a very easy question, right? Long stature as in long height, broad palm, furrowed tongue, retardation of mental development. Which is the thing that they do not show? That is a question. And please, everyone who said A, kudos to children who are saying yeah, A, because this is the answer. Yeah. So long stature is not something that is found in Down syndrome. They have short stature. Okay, kudos to all of you. I'm going ahead with the next question. Okay, otherwise we might not be able to finish. Now the gene for hemophilia is located on X chromosome, right? Gene for hemophilia is located on the X chromosome. That is why it is an X-linked recessive disorder. Now hence it is normally impossible. It is normally impossible for what? Hemophiliac father to pass the gene to his daughter. For example, I am saying hemophilia gene is located on the X. Father has one X and one Y. So basically, is it impossible for the hemophiliac father to send this gene to a daughter? No, my dear children. To a daughter, he will send this particular gene because daughters are having XX chromosome, right? So one they get from the mother, one they will get from the father. So this is wrong. It is not impossible. Now carrier mother to pass the gene to her daughter. Carrier mother, my dear children, suppose this is a carrier mother. Can't she pass this gene to her daughter? Yes, she can. She will definitely pass either of the X. So this is also possible. Carrier mother to pass the gene to her son. Okay. Carrier mother to pass the gene to her son. Isn't that possible? Again, that is possible because in the case of a boy, a son to be formed, you get X from the mother and Y from the father. So again, this is possible. Now, the children who got your answers as D, you are correct. Hemophiliac father to pass the gene to his son. Array, to the son, can he pass X chromosome? No, he can only give Y chromosome. X chromosome comes from the mother. So can a hemophiliac father pass this gene to a son? The answer is no, this is impossible. I hope you're enjoying this. Yes? So great of you. Let me read out the names and then you go ahead with the next question. Okay, read out the next question. Let me tell the names. Oh my god, so many people out there giving the correct answer. So good. Children, I'm so sorry I cannot read out the names. It's too long list, but I'm just reading out here and there from somewhere. Mehek, CR Shetty, Kirtana, Monisha, Kiki, J student, Angel Gaming, Nikita. Okay, and AK, every one of you, my dear children, all of you are doing really, really great. Yes, all of you who got the answers correct. Ma'am, you are just really Jasmine for biology. I hope. Okay, thank you so much. RX fans, am I audible? I hope I'm audible. Yes, so others have started answering for the next thing. Oh my god, now I'm in tension. Who among the following use the frequency of recombination between gene pairs on the same chromosome as a measure of distance between genes and map their position on the chromosome? Who was it? Let me see who was it. So everyone is saying, someone is saying D. So B, let me see who also saying B. Your answer is B. Your answer is B. His name was Alfred Sturtevant. Okay, and he was a student of T.H. Morgan. Let me just explain Chuttu thing. Okay, so basically over here you have a chromosome. Okay, and suppose there are gene A and B. If they are located far away, what is happening? There is higher chances for recombination. As in, this gene can move from this side and go to another chromosome right over there. Alone. Okay, it can move out there alone and leave behind this A gene over here. So don't you find a new combination, a chromosome having B and a chromosome having A over here? This is new combination. So basically these genes are loosely linked. What do you call this? Loosely linked. And loosely linked genes will be having higher recombination. Always remember, I'm telling it 10,000 times because it is very important. Loosely linked genes, higher recombination. That means you and your best friend. For example, I'm taking two people, okay? Monisha and Priyadarshini are very close to each other. They are best friends, okay? Now what will happen? If they are best friends, there are least chances that they will include someone else in their group. So there is less recombination. That means if they are closely linked, that means less recombination. But suppose Monisha and Priyadarshini are not that great friends, there are higher chances that new combination can come in. So here also, if they are loosely linked, more recombination can happen. So Alfred Sturtevant just said this, that basically if I am able to calculate the recombination, if it is more, that means probably they are loosely linked, they are far away from each other, and in that way I can locate where are the genes. That is known as gene mapping. Okay, done? Bhagat, 2 chapters. Okay, I am doing principles of inheritance and variation and molecular basis of inheritance. I hope this is clear. Okay, going ahead with the next question that we have. Children, all of you got it clear? Give me a heart, give me a thumbs up, anything, so that I get to understand. On all of you who said B as the correct answer, I see certain new people also out there, Komal, and then Bhagat, all of you are there. Great, great, great children. Your mic is not working. Yashu, children, isn't my mic not working? Am I not audible? Can you please tell me if I am audible? Just tell me I am audible. If I am not, tell me that I am not audible. Oh, good. Yes, I see so many hearts flying around there. Okay, great, great, great. Can we solve this question? Thank you, SSLC. Thank you for telling me that. Okay. Yes, you're. So those of you who feel that I'm not audible, can you please just recheck once? Okay, can you reload once? Can you just check your connections? Yeah. Now, increase in percentage of recombinants for a pair of characteristics indicates. Now tell me the answer for this. I just taught you right away. Okay, Pradeep says 11D. RX fan says D. Let me see who all got to understand. Kirtana says D. Okay, let's see. So it's saying percentage, increase in percentage of recombination. That means they are loosely linked. They are loosely linked. If they are loosely linked, they are placed far away from each other. They are far away from each other. So they are tightly linked? No, wrong answer. So both A and B is correct. That means D is correct. And you all got to understood what I tried to speak in the previous slide. Great. Awesome. Okay, so your answer is A and B together. That is D. C.R. Shetty, great, great, great. SSLC Gaming, Mithun, Suvidha, Nikita, everyone, Priyadarshini, Chandrasekhar, Angel Gaming, Baswesraj, everyone, great, great, great. Now, question number 12. Question number 12, number of different types of genotypes and phenotypes are possible in inheritance of ABO blood group in humans are. Children, ABO blood groups in humans are. What will be the answer for this? Number of genotypes and phenotypes. Phenotypes means physical appearance. Okay. So, in ABO blood group, how many different types of blood group is there? A, B, O and AB. A, B, O and AB. Right. So, how many phenotypes? Physical appearance? 4. Okay. 4 in number. So, basically the one has 6 and 6. We'll cross it off. This is wrong and this is wrong. Now, we have 2 options. Genotype as... okay. So, someone is saying C as an option. Is it true? Can you just check? You're talking about genotype. So for A blood group, how many genotypes? It can be IA, or it can be IA, IO, right? Now for B blood group, IB, or it can be IB, IO, because B is dominant over O, here A is dominant over O. We got already 4. Now I'm going ahead. For O blood group, you have IO and IO. For AB blood group, what do I have? I have IA and IB. So 4, 5 and 6. Your answer should be D. Your answer should be D. Exactly. So those of you who got your answers at D, you are absolutely correct. Okay. Manjula and then, oh my God, just a second. Okay. Manjula, Karna, Pratna, Komal. Okay, I'm going up. Great, 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 my dear children. Nikita, children. Awesome. So do not forget this. Okay. Going ahead with the next question. Mother is homozygous B and father is heterozygous A. What will be the possible blood group in their progeny? Take out your pens, try to solve it. And all of you who got your answers correct, RX fans, I'm reading out the name, okay? Sandeep Patil, Komal, C.R. Shetty, Priyadarshini, Nikita, Rashid, and Mithun, Yashu, Baswesraj. Why is my voice not clear? JE student, everyone. Okay, SSLC Gaming, Manjula. Children, I feel so happy that you are trying. Try out the 13th question. Bacha, mother is homozygous for B. That means it is IB. Cool. And father is what? Heterozygous A. Heterozygous A means IA and IO. Cool. Now you have to tell me what will be the possible blood group in their progeny. So B goes with A, I get IAIB, that means AB blood group. B goes with O, I get IBIO, that means B blood group because B is dominant over O, right? B goes with A, again co-dominancy IAIB, I get AB blood group. and B goes with O, again I get IBIO, that means B blood group. So I can see only two type of blood groups, AB and B. So what will be your answer? Your answer will be 13A. 13A. Who said B? Who said B? It is 13A. Great. Great, 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 great. Okay, it's 13A. Please make it correct. Okay, if you are worried about anything, try to solve it again and just check it. Don't do the mistakes on the day of your examination. It is okay to make mistakes over here because this is how we learn, right? So totally fine. It's A. Okay, I see so many hearts out there. Yes. Now question number 14. When a tall plant with round seeds crossed with a dwarf plant with wrinkled seeds. Okay, fine. The F1 generation consists of all tall plants with round seeds. All tall plants with round seeds. Why? Because tall and round are dominant, which is why F1 will be, F1 generation obviously it will be tall and round only. The dominant character will be shown up. Now what will be the proportion of dwarf plant with wrinkled seeds? That means you are asking about this thing. You are asked about this thing. In what? F2 generation. In F2 generation, what will be your answer? So I see the answer. Children, can you please put down like this 14 ABCD so that I understand what question here. I would be saying, yeah, B is correct, C is correct, and then you might be asking about different question. So I don't want that to happen. So please tell me 14 you're talking about. Okay. Someone says D, 14D. Rashid says, C.R. Shetty says 14D, Chandrasekhar 14D. Yes, the answer is D. How do I calculate it easily? So in F2 generation, in a dihybrid cross, when two characters are there, that is tall and round, two characters are there, right? In F2 generation, we get 9:2, 3:2, 3:1 ratio. Always remember, 9 of them will be the dominant character. Dominant over here is what? Tall and round. And one will be obviously the recessive plant. Okay, so recessive plant is what? Dwarf and wrinkled. Dwarf and wrinkled. Okay, now if you are asked about 3 and 3, okay, what will be them? Can you tell me? One will be tall, and instead of being round, it will be what? Wrinkled. We are going to make new combination. Here it was dwarf, but this time with wrinkle, it won't be wrinkled, it will be round. But you are asked about dwarf plant with wrinkled seed, it will be only one. One out of what? 9 plus 3 plus 3 plus 1 is 16. So 1 out of 16. So this becomes your answer. Okay? Okay? Great. Appa, my head. Fine. So all of you who got D as the correct answer, kudos to you. I hope children you are understanding. Not understand? Yes, sure, I'll make you understand. Listen, always remember in F2 generation the ratio is 9:3:3:1. Okay, now what will be 9? The first parent that we took, the dominant parent. So dominant parent over here is tall plant with round seeds. So tall and round will be 9 of them. Okay, now one of them is going to be dwarf plant with wrinkle seeds, the recessive parent. Okay, now in middle you would be having recombinants, new combination, my dear children. There tall character will be there, and instead of round it will be wrinkled. New combinations there. Here dwarf will be there, but along with it, it will be round. So these are the new combinations that we find. These are the older combinations that we had. You are asked about what is the proportion of dwarf plant with wrinkle seeds, that is the recessive parent. So over here it will be 1 out of what? Out of 16 of them. So 1 out of 16 is the answer. What does kudos means? Kudos means great, awesome. Okay? Okay, Ajay, hi my dear child. I hope you got it now, all of you. And going ahead with the next question, which of the following statement regarding sickle cell anemia is not correct? Whenever this type of question come, underline what is not correct. You are asked about incorrect statement. Try to find it out and let me talk about all the others who are giving their 100% efforts into answering. So children, I do not know whether you answered correct or not, but I am just reading out the names of the children. Okay, Karna, Baswasraj, Mithun, Preeti, Komal, Anusha, Rashid, RX Fan, Chandrasekhar, C.R. Shetty, oh my god, Manjula, Priyadarshini, Baswasraj and Kiki, Sneha, Angel Gaming, Suvidha, Chikegowda, Ajay, Yashu, every one of you children, Mehek, I see someone new, and Angeline over there. Okay, all of you great going champions. So what is not correct? So caused by substitution of glutamic acid by valine in the beta globin chain of hemoglobin. Yes, it is correct. Okay, sickle cell anemia is caused because of a mutation that is happening at the 6th codon, my dear children, where instead of having GAG, which was coding for glutamic acid, now it has been replaced to GUG, which is coding for valine. How do you remember this, ma'am? Remember, A comes before V. So initially we had A, then you have U. Okay, now G comes before V, so glutamic acid was the initial one, then it was replaced by valine. This is happening in the beta globin. These codons are going to code for what? Beta globin chain. And this is the 6th codon. So this is correct. Controlled by a single pair of alleles HBA and HBS. That is also correct. Characterized by elongated sickle-like RBCs with a nucleus. Yes. The after effect is what? RBC becomes like sickle shaped. An autosome linked dominant trait. Wrong. It is what? Autosome linked recessive disorder. It is recessive disorder. This is a wrong statement. So your answer becomes D, and whoever got it D, awesome. Kudos. I'm telling kudos again and again. That's how I am built, I think so. I'm so sorry if it's offending you. But children, you're going absolutely great. I'm so happy that you are trying it out really well. You are there. Almost 48 of them are in live. That's so super good. Yes, I see Yogita, Manjula, all of you, Suvidha, Monisha, Kiki, Siya Shetty. Try it out. Next question. Nikita. Ma'am, when PUC result? PUC result could be coming out soon, anytime. Even we do not know the dates. Telling a wrong date would be a wrong information. I do not want to misguide you. Yes? So, which one of the following conditions correctly describes the manner of determining the sex? Correctly defines. Okay. Can you try it out? 16th question. Chandan, someone new is here. Children, I hope you're liking the session. Am I going fast? I hope I'm not going fast. Okay, so a little bit fast is necessary over here because we have so many questions to complete, but yeah. So, hi Sharda, 16B. Someone says 16, answer is B. Okay, let me see. Everything is smooth. Okay, Kiki, thank you. Swift is there. Hi Swift. So, 16B again. I see B and C and D. Okay, B and D mostly. J student Bhumi, hi. Riyas, Tehsildar, B. Okay, Lokesh says B. So let me see. You are asked about correctly defined. So homozygous sex chromosome ZZ determine the female sex in birds? No. If the bird has to be female, it has to be ZW. It is not ZZ. ZZ is for males. It is the only one that shows female heterogamety. Two type of gametes are being produced by birds, female birds over here. Either Z or W. Only example of female heterogamety. The wrong statement. XO type of sex determination determines male sex in grasshopper. Yes, in insects it is XO. That means there will be X chromosome or there will be no chromosome, which is why it is XO. So whenever you have a combination like this, this will turn on to a male grasshopper. So this is entirely correct. So all of you got BS. Correct answer. Kudos. Great. Now, XO condition in humans as found to Turner syndrome determines male sex. No, my dear children. Okay. It will be a female but with a masculine body. Now, homozygous sex chromosome XX produce male in Drosophila. Array. No, never. Drosophila, human being, same. Okay. XX means it has to be female, not a male. So, this is wrong. So, only correct statement is B. Booming. Thank you so much. Okay, so Suvidha, Chandrasekhar, Rashid, Muslim, Siya Shetty, all of you, my dear children, awesome. B is your answer. Okay, so sorry children if I'm missing out certain children's name. Okay, I would be calling them out. I'm randomly calling you guys. I want to include as many people as I can. Okay, and I'll try to remember your names as well. So question number 17. When both alleles of a pair are fully expressed in a heterozygote, they are called heterozygote. Means basically they are different. For example, A and B. This is heterozygote, and both of them are fully expressed. What do you call this condition as? This is an easy question, right? 17B. Charlotte is there. Hi, Charlotte. Okay, Charlotte, Siya Shetty, Bhumi, Chandrasekhar, Lakhita, Riyaz, Mithun, Narasimha, Chandan, Yashu. Oh my God, all of you are telling the correct answer. Rashid, Bhumi, Kiki, Nikita. Okay, Monisha, Chikkagowda. Okay. Rashid again. Children, yes, it will be codominance and it is shown in blood group. What blood group? ABO blood group, where A and B both are dominant. Okay, which is why they are expressing fully expressed. If the question was, it is not fully expressed, your answer becomes incomplete dominance. Okay, so over here it is fully expressed, so your answer is this. Yes, ABO blood group, Kiki says. Great. Now question number 18. This I want to see who all get me the answer. Okay, be ready for question number 18. Have your pen ready. If both parents are carriers for thalassemia, both of them are not affected, they are carriers for thalassemia, which is an autosomal recessive disorder, what are the chances of pregnancy resulting in an affected child? That means the child has thalassemia, it's not a carrier. Can you tell me the answer for this? This is 18th question. Okay, Angelina so fast with 25. Okay, Bhumi says D. Chandrasekhar says B. Okay, Manusha says D. CR. Everyone, please try it out. Do not just randomly say, have your pen. Try it out once and tell me. So basically, it's like this. Okay, both the parents basically. Okay, suppose I'm just writing randomly, okay. This is A is normal and T is the one that is affected. A is normal, T is the one that is affected. That means they are carriers. They are what? Carriers, my dear children. Now we have to find the one that will be having both of this together. So this and this goes, I said HBA and HBA. It's a normal child, okay. This and this moves, I get HBA and HBT. Again, it's a carrier, not affected. This and this goes again, HBA and HBT. Again, just a carrier. This and this goes, I get HBT and HBT, and here comes the affected child. So all of this together, if I say it's 100%. So what is this? 100 divided by 4 is 25%. All of them will be 25, 25, 25 each. So this is, my dear children, 25%. This will be 25%. So your answer becomes B. Your answer becomes B. Mithun Baba, can I check once? Okay. It is B. Cool, got it. Now, I am reframing the question and I am asking you, listen to me carefully. This is a whole new question. Okay. Among them, how much percentage would be carriers? How much percentage of them would be carriers? Can you tell me? Your answer should be given like this. Optional. Optional is equals to ABCD. Optional is equals to ABCD. Yes. Chandrasekhar says 50. Angeline says 50. Angel Gaming said, then it's 100. Carrier. You see, carrier. 2 of them are carriers. Only 2. So 25% plus 25% becomes what? 50%. Your answer would be what? 50% then. Got it? Listen, this is carrier, this is carrier, this is affected, this is normal child. Okay, so 25, 25, it will be 50%. Please get that correct. Okay, it happens. It happens. It happens. Here, Shati, it's totally fine. Okay, so that is something that we have to remember. Chalo. Sharana Baswa Ambigar, I hope, Baba, you got that correct. Okay, children. Okay, question number 19. After this, we'll take a, no, after this. There are 5 more questions. After that, we'll take a short break, chutu break. Okay. Now, which of the following best describes polygenic inheritance? So, polygenic inheritance, I'll tell you. What does that mean? That means one trait is being basically shown by or basically, for example, I have one trait, one character. What is this one character is decided by multiple genes? Gene A, B, C, this is known as polygenic traits. Okay, so now you tell me the answer. And Angeline says, ma'am, I need a help. I didn't start to study physics and chemistry. You can still start. You have some time left. Okay, so Aishwarya ma'am gave a video on strategy. You can watch this video. Okay, and please start, prioritizing PU2 chapters now. Okay, PU1 chapters we can do it later, PU2 chapters, prioritize it, solve as many questions as you can because most of the questions are going to come from PU2. Last minute if you have some time left, you can go ahead with PU1, but don't take the risk of leaving chapters of PU2. You will be able to do it, Angeline. You are going to do really, really great. Okay, so all the children who are here and who are feeling that I have not reached anywhere, there is a lot of things left. Baba, you have reached until now, you have started. That is why you are here in this particular live, and I'm so proud of you guys. You are doing so great, and you should be doing like this. You should be going on like this. You are going to crack KCET. Okay, so do not worry about anything. Now, I see Kiki saying answer is polygenic trait is human colored skin. Okay, let me see. And all of you saying D. Let me see. So, ABO blood group in human and flower color of Mirabilis jalapa. Flower color of Mirabilis jalapa, that is strap diagon, is showing incomplete dominance, and abelian blood group is basically complete dominance, so this is not the thing. Hair pigment of mouse and tongue rolling in humans, no. Human eye color and sickle cell anemia, no. Human height and color of skin, yes. Which is why we have multiple skin colors. My skin color is not your skin color, right? Everyone has different skin colors because basically it is coded by multiple genes. For example, I have two genes like this, then there is this, then there is this. That is why I have got a skin color. Yours might be A, B, B, C, C. Either way. So there are multiple genes coding for one thing. That is why this is different. So your answer becomes D. Your answer becomes D. Ma'am, please could you explain height is also an example of polygenic. Polygenic, okay? polygenic. So why height is also similarly. I'll just give me 1 minute, children, for explaining this thing. Very important, that's why. So basically, suppose, okay, over here I have AA gene, BB, CC. In that case, my genotype is AA, BB, CC. Okay, so basically the pigment that is going to be produced is very much, which is why my skin color will be very dark. On the other hand, over here, if it is small a, over here it is small b, that means this pigment, the protein, amount of protein that is going to be produced is going to be less, which is why the pigment, my dear children, if you see the genotype now, it will be different from what is there over here. Similarly, for height also, you have multiple genes. Okay, for example, if there is lesser protein being produced, the growth hormone which is there, that is also not going to be produced very much, which is why the height will be less comparatively. Okay, if I have all the big dominant ones, it will be more, so height will be more. I hope you got it now. Okay, children. So height and skin, both of them are example of polygenic inheritance. Am I clear with this? Can you give me a thumbs up or a heart if you understood this? If you still didn't understand, I am here to tell you. Okay, Bhagat, thank you so much, means a lot. My favorite bio queen. Oh my god, that's a big thing. So yes, I see your hearts coming and rolling around, so I feel it's correct. So you got to understand. Children, start with 20th question. A colorblind man marries the daughter of another colorblind man whose wife had a normal genotype for color vision. In their progeny, what will be happening? Can you please try to find out this? Okay, and now let me see the children who are answering. I am already constantly checking that. So Monisha is being sad. Baba, why are you sad? Okay, previously I saw you were sad. Now I hope you are happy. Bhumi, Mehek, Bhagat, Mithun, Bhumi, Suvidha, Chandan, Jay, Chikagowda, Siyar Shetty, Sharna Baswa, Angel Gaming, Lokesh, Angeline, Lakita, okay, Nikhil, Mehek, Shalit, okay, Monisha, Priyadarshini, everyone, Jay, all of you. I hope you're doing good. Yes, yes. Chalo. So over here, a colorblind man, that means it is going to be like this. Fine. Marries the daughter of another colorblind man. Okay. So another colorblind, this is another scenario, okay. Another colorblind man, that means like this. Whose wife had a normal genotype for color vision. That means the wife is normal. Now we have to find what will be the daughter. So daughter can be just like this. Daughter can be a carrier, my dear children. Daughter can be a carrier, or basically, yeah, daughter have to be a carrier. There is no other option because the father is having the color blindness. So anyway, the father will be giving this particular gene, which is why both the daughters will be colorblind. Now over here, I have to make them mate. So the mother is, my dear children, carrier female, and the father is having color blindness. That is what we understood from this. In their progeny, what is the thing that you would be finding? Let's make the cross first. So this and this goat, you get XC, XC, that means colorblind female. This and this goes, you get XCX, that means this is a carrier color blindness. And this and this goes, you get XCY, that means colorblind male. This and this goes, you get XY, that means normal boy. Now, what is the thing that you can, all children would be colorblind? Wrong. Not all children are colorblind. Only this female and this boy is colorblind. All their sons are colorblind. Are all the sons colorblind? No. One is normal and one is colorblind. Wrong. None of the daughters would be colorblind. Array, one daughter is already colorblind, so that is also wrong. Half of their sons, that means if they have two sons, half of their sons would be colorblind, yes, and half of their daughters, as you can see over here, would be colorblind. Do not count this as colorblindness, this is just a carrier. Okay, so she will also be colorblind. So your answer becomes D. Okay, so your answer becomes D. I am Rekha. Sharda says I am Rekha. Okay, you're Rekha. I'll remember that. So you are Daniel. All of you who got your answers correct, kudos to you. Okay, great going, children. That means you're understanding things. See so many hearts also. Thank you for the hearts, everything. Now study the pedigree chart given below. Now the same pedigree chart we studied initially. Okay, try to solve this question ASAP, and I'll read out the names of the children who got the answers correct. So 21st question, try it out. So let me see who got it correct. Okay, Rekha, I will be explaining you the law of segregation towards the end. Will that work for you? Okay. Charlotte, yes, PDF will be given in the channel, WhatsApp channel, so join the WhatsApp channel, link in the description. Nikhil, Muslim, and Chandan, Monisha, UI Daniel, Akash, Riyaz, Rashid, Chandrasekhar, CRI Setti, UI Daniel, oh my god, I'm not able to read out the names. Anusha, Priyadarshini, Subhida, all of it. CR Shetty, great. Okay. Can you, ma'am, I am Pragati, can you explain again? I didn't understand. I'm so sorry, I saw it right now. I hope you didn't understand this. So listen, here a colorblind man marries the daughter of another colorblind man. So colorblind man, we have to already make it. Now we have to find what is a daughter. So daughter, for finding a daughter, we are going in this frame. Okay. So what is about the daughter given? That the daughter of another colorblind man, that means the father was colorblind, whose wife had a normal genotype. So wife is normal. So whatever daughters you try to make over here will get 1x from the father. So that means it will be a carrier. She will be definitely a carrier. Now I'm going to place it over here, and I'm going to try to find out. So when I tried to find out, I found out that in 2 females, one of them was having the color blindness and one was the carrier. In 2 males that were produced, one of them had the color blindness, other was normal. So can I say half of their sons? Okay, in between 2, only one had. So half of the sons and half of the daughter would be colorblind. I hope now you might have understood this. Yes. Now, children, please try to solve this. Pragati, I hope you understood that, Baba. What does this mean? Inheritance. Okay, 21A. Someone says 21A. Inheritance of recessive sex-linked disease like hemophilia. So someone who all said A, you didn't understand this particular pedigree chart yet. That is what I would make you understand. So listen to me carefully. Whenever you are skipping generation, my dear children, whenever you're skipping generation, it has to be recessive disorder. Okay, so here parents do not have it, but the children have it. Here parents do not have it, but the children have it. That means they are carriers. That means they are carriers. They have one gene affected. If both of the genes are affected, then only they will have it. So that is why I'll say it is a recessive one. Now, again, listen to me. If the boy and the girl child are equally affected here and there, my dear children, it cannot be a sex-linked disorder. It will be autosomal. It is not a disorder in the sex chromosomes. It is an autosomal. Here also you can see girl and boy, both of them are affected. So it is autosomal, not sex-linked. Now over here, inheritance of recessive sex-linked disease like hemophilia will be wrong because it is sex-linked. Inheritance of a sex-linked inborn error of metabolism like phenylketonuria, wrong, because phenylketonuria, my dear children, is not a sex-linked disorder, it is an autosomal disorder, wrong. Inheritance of a condition like phenylketonuria as an autosomal recessive trait, yes, because it is phenylketonuria and it is autosomal recessive trait that is being shown over here. So your answer becomes C, it's not D, okay. Yes, it is an example of sickle cell anemia, but sickle cell anemia is also autosomal recessive disorder. And what? Phenylketonuria is also autosomal recessive disorder. You get it? So pedigree chart can come for anything. Okay, so which is why your answer is C. Oh God, oh my God. So children are thinking of God now, and that's so great, so good. But it's okay. At least now you understood this. So your answer is C. Okay? All of you who got C as the option, kudos to you. Come back to this question, write this question number down, come back, revisit this question after the live, okay? Superb. Now, question number 22, pleiotropic gene is what? Try to answer this. Got it, ma'am? Thank you, Kiki. Okay, let me tell all the children who got the answers correct. Okay, I'm just reading out the name. I do not know if you got the answer correct or not. Akash, Leela, Nikita, Angeline, Narasimha, Nijagana Aradhya, Priyadarshini, Muslim, Leela, Suvida, Riyaz, Devraj, CR Shetty. Okay, thank you all for basically at least attempting the question, and all of you got it correct. Kudos to you. So Devraj is with an F option. F option is not there sadly. So pleiotropic gene inhibits crossing over, promotes crossing over, controls only one phenotype, controls several phenotypes. What will be the answer for this? Controls several phenotype. Your answer will be D. Straight question. Pleiotropic. So polygeny means multiple genes for one character. Pleiotropy is the ulta. One gene for multiple traits. One gene for multiple traits. So this controls several phenotypes. Okay, so D becomes your answer. Can anyone give me an example of this? Pleiotropy. Any disorder? Any disorder? Explain every option, ma'am. Inhibits crossing over No, my dear, it's not happening. Okay, it does not have anything to do with crossing over. Promotes crossing over? No. Crossing over means the chromosomes are here, they are crossing over, they are exchanging the things. That is crossing over. That does not have anything to do over here. Controls only one phenotype. That means only one character. No. It is controlling two character. Okay, so your answer is yes, phenylketonuria. Can I just take a second to explain this for the children who didn't understand? Okay, so listen to me carefully, very carefully. So there is a disorder that is phenylketonuria, where what is happening? There is an enzyme, phenylalanine hydroxylase. It is not being produced. So which is why the phenylalanine which is there in our body, usually it is converted to tyrosine. Usually it is converted to tyrosine, but since this enzyme is not there, this conversion is not taking place, which is why phenylalanine accumulates as phenylpyruvic acid. It accumulates as phenylpyruvic acid that will be causing mental retardation. So this is one trait that it controls. It will be leading to mental retardation. Now, second trait that is going to be controlled by the same gene is that since tyrosine is not being produced, tyrosine is an essential element in the production of melanin. That is giving the skin color, hair color and things like that. So since tyrosine is not being produced, what will happen? You will be finding reduced skin pigmentation and hair pigmentation. So this is the second trait that the same gene is controlling. The same gene coding for the enzyme is controlling two things, which is why I say pleiotropic genes are the one that control several phenotypes. This becomes your answer. Okay, so reduced skin pigmentation, hypopigmentation you call it. Cool? Yes, you are Daniel saying something. I am going to read it out, and question number 23, you read that out until then. Mendel's law of independent assortment is applicable for what? So, you are Daniel says pleiotropic is same as the leader. As the leader controls many people, then pleiotropy controls several phenotypes. Yes, exactly. Great. That's awesome. Okay. Leela, please fast. I cannot go faster than this. Yes, there are certain children who do not get it, which is why I stop at time. I'm so sorry if that's causing any trouble, but yeah, I have to make them understand as well. So please try. Mendel's law of independent assortment is applicable for 23C. Yeah. Okay, let me see. All linked genes only, all genes in all organism, all genes for pea plant only, non-linked genes only. What does law of independent assortment says? Law of independent assortment says that, for example, Jasmine and Ashwarya ma'am, both of them are not going to go together always. So one will go in this direction, another will go in that direction. That is law of independent assortment. They are not dependent on each other. Okay. Now, for example, if we were so close, we would be moving together. And that will be what say not performing independent assortment. I'll say it is opposite to independent assortment. We are dependent. So basically, law of independent assortment will be applicable for what? The genes which are far away from each other. The genes which are far away from each other, if they are so close, they will be moving on together itself. So all genes only wrong. All genes in all organisms wrong. All genes of pea plants no. Non-linked genes, that means they have to be far. Then only law of independent assortment can be working. Okay, so your answer becomes D, not A. It will be D. Okay, independent assortment means the genes are not going to be together always, they are going to move away separately, independently, and for that the genes have to be non-linked. They do not have to be close, they have to be dur. So your answer becomes D. Got it? Children who got to understand this, give me a heart or a thumbs up, because that is a question that needs attention. Okay, some children might not get it, which is why I am telling you. Yashu Sada. So okay, try this. Okay, I am seeing few hearts out there, 100. Okay, thank you. A test cross distinguish between what? The question that we already did. Can you try it out, please? Okay, so now Leela said the correct answer, previous 23. Yeah. Wait. Everyone who got the answer as D, you are getting the answer as correct. Otherwise, the answer is wrong, but it's okay. So Nikita, Leela, Jai, and Chandan, Priyadarshini, Rashid, UI, Daniel, Devraj, Nijaganaradya, Chandan, Angeline, Nikita, all of you, thank you for trying it out. Okay, so happy that you guys tried it out. Okay, 24C. Okay, test cross distinguishes between two homozygous form, two heterozygous form. Two homozygous means, for example, this and this. This is homozygous. Two heterozygous means this and this. Why do I need to distinguish? A homozygous recessive and a heterozygous form. A homozygous recessive and a heterozygous form. Is it for distinguishing that a homozygous dominant and a heterozygous form. This would be correct. I want to distinguish between this. This is why I do a test cross. I hope you are able to understand this. Okay. Your answer is D. Children, all of you got it. Homozygous dominant and heterozygous joint. Nijaganaradhya, Baba, that's not C, it's D. Children, my voice is low. Is it low? No problem in voice. Okay, thank you, Leela. Ma'am, how? Okay, I'll tell you. Listen to me carefully. Okay, test cross is being a problem for all. So, capital T, parent. Okay, capital T, small t, parent. This is homozygous dominant. This is heterozygous. I am doing a test cross. What is test cross? Where basically I am taking a plant and trying to mate it with a recessive parent like this. Here also I am going to do the same. And now when I do it, I find that this and this goes capital T small t, this and this goes capital T small t, this and this goes capital T small t, this and this goes capital T small t. Oh my God, all of them are tall. All of them are tall. That means this is homozygous. You will get to understand why. Because when I cross this, I will find this and this capital T small t, but here I find small t and small t. So I find dwarf plants as well along with tall plants. And do I want that? If I am someone who is wanting tall plants in all the generation, can I be okay with this? No. This is why I do a test cross to find whether it is homozygous or heterozygous, so that I can just take the homozygous and use for my crop breeding programs. Okay? Okay? So, I hope that is understood. Test cross is between F2 generation and double recessive parent. So, it necessarily actually, okay, it's when you do not have to, it's not F2, F1 generation in general, okay, it's just to find whether it is homozygous or heterozygous. Obviously, you generally say it is F2, that will be more correct statement to say that, because once you get the progeny and then you go back, do the test cross, that will be more correct. So, F2 would be more correct, but in this way also if the question comes, in the question if it comes, it is correct. Okay. So, your answer would be D. At least I hope you got it. Children, you got it? Give me a heart or thumbs up if you got it. Hi, Black Havoc, Devraj, Priyadarshini, Narasimha. All the other option anyways, it does not mean anything. So, you just have one option. Am I audible? Yes, got it. Sonia Naik is there. Okay. Okay, thank you children for acknowledging. And someone had a doubt. Who was that? Shriya Shetty? Baba, I hope your doubt is also cleared with this. Always remember, test cross is you are crossing with a recessive parent. Now, last question of of principles of inheritance and variation. That is this. Sayanath. Okay, identify the correct match between inheritance and their examples. 25. Try to find it out. Okay, let me read out the names of the children who are there. V Master, Guru Swami, I see so lots of new people are there. And then our old children, Nikita, Nijaganaradhya, Nikhil, Yashu and Jai, Devraj, all of you great going, all of you champions. Okay, Spurti is there, Priyadarshini is there, Varsha, hi. Big hi. Sayanath, even though I'm from CS, Bio will be my favorite subject. Okay, thank you for that, but I hope you studied CS also. Okay, anyways. So 25, Priyadarshini says it's C, and Siya Shetty says it's D. D and C. Can you choose one? Adita, Akash, hi. Love your teaching. Thank you so much, Spurti. Children, can you choose between the option? So 25B. So I have to find the answer, right? Complete dominance. What do you mean by complete dominance? Let me try to find the answer. Complete dominance is when, for example, I have capital T, Ayyo. I have capital T, small t. This is completely dominant, which is why it is tall. This is complete dominant. So where do I find it? In flower color in P, the flower color of the P. Okay, you have violet and white. White is always dominant over white. Okay, sorry, violet is always dominant over white. So A goes with 4. A goes with 4. I just have one option, and with that one option I can find all the answers, right? So now multiple alleles. Multiple alleles, where do you find it? In blood group. That's the most common thing that I remember. Blood group, I have A, B, O, all of them are multiple alleles. So B goes with 3, correct. Co-dominance, that means both of them are equally dominant. Again, it can go for blood group only. But since blood group is already gone, I have one more option, that is coat color of cattle. Now if you're someone who's not sure about this, okay, let's leave it like that. Let's go with the D option, incomplete dominance. That is shown by what? Flower color in snapdragon, right? Where what is happening? You have capital R, small r, but this capital R is not completely dominant over small r, which is why instead of giving red color, it is pink color. It is what? Pink color. So I'm pretty sure about that. So hence, what is left? Co-dominance is left and coat color is left. So C goes with 2. Okay. So your answer will be A. Who all got it as A? Who all got the answer as A? Hi ma'am, new to the class. Okay, I welcome you to the class. Okay, it's A. Who all got it as A? Got it correct. Who all got it as B, C, D? It's wrong, but it's okay. We are going to rectify it. Okay, cool. Chalo. And with that, we complete all the questions from principles of inheritance and variations. Okay. Now, I hope everyone got all the things correct. I'll request you to have your water bottle, drink some water because I also need some water. Okay, I'll also drink some water, you guys drink some water, and we'll start off with the next chapter, that is principles of inheritance and variation. If you're liking this particular video, do not forget to like the video in the live and also put down your comments if you have some doubts or if you think that this particular video is useful for you. Either way. So I see so many hearts and things like that flowing around there. Children, please drink water, hydrate yourself. Fast. Okay. Yes, ma'am. Please stay hydrated. Thank you so much, Kiki. Continue live section. Okay. Okay. Let's get ahead to the next chapter. Principles of inheritance and variation is getting completed. Going ahead with molecular basis of inheritance. Hi Ravi Kumar. Correct time to enter the class because we are starting off molecular basis of inheritance. Ma'am, I didn't understand biotechnology lesson. I had made up a video for biotechnology, that is master stroke. Go watch it. It's very nice. I think so. Okay, you can just watch it once. It will be helpful for you. Okay, and also top 30 in top 30 also have covered that. So please watch those videos. It is easy chapter. It's fun chapter. Chalo. Who among the following presented evidence for DNA to be the genetic material? 1D. So let's see who all got this particular chapter clear. Varsha, my name is Jasmine Joy. Okay, you can call me JJ ma'am. No problem. You don't have to call the entire name. Okay, now who among the following presented evidence for DNA to be genetic material? My dear children, all of you are saying D. Let me see. Nijaganaradhe, Chandrasekhar, Kizar, and then Devraj, Nikhil, Suvida, Kiki. Okay. Children, Jyoti, I see new people out there. Great. All of these guys, my dear children, all of them said that basically DNA is going to be acting like a genetic material, because Avery, MacLeod and McCarthy did an experiment where what did they do? They treated the genetic material with RNAs. That means they tried to kill the RNA, but still transformation was taking place. Okay, they tried to put protease, still transformation was taking place. But then when they treated the genetic material with DNAs, the transformation was not taking place, and then they got to know that basically DNA is a genetic material. So all of this is correct. Avery, MacLeod and McCarthy. Going ahead with the next question. 2. A nitrogen base linked with a pentose sugar at carbon number 1 forms what? Yes, tell me the answer for this. Guruswami, Shalit, Varsha, Bindu and Muslim, Priyadarshini, Chandan, all of you, great. Just finished 2-hour study session, but hey, can't leave now. Oh my god. Thanks. Okay, so basically my dear children, you have a pentose sugar like this. Pentose means 5 carbon sugar. 1, 2, 3, 4 and 5th carbon is outside because it wants to be outside. Now on the first carbon you have what? A nitrogen base, NB, nitrogen base located. What do you call this structure as? Nucleoside, nucleotide, polynucleotide, none of this. Okay, let me see. 2, okay, wait. Chandrasekhar 2A, okay. 2A, Kizar said A, Jyoti said 1, okay. Nijaganaradhya A, Angeline D. Varsha A, Chandrasekhar, Sudeep A, Charlotte A, Gagan A. Hi Gagan. Okay, Preeti, Bindu, all of you A. Monisha, that's so sweet of you. Okay, so your answer is what? A, my dear children. This structure is known as nucleoside. Now, I'll tell you one thing. On the 5th carbon, when the phosphate also comes and attaches, the whole name changes. Now, it is not nucleoside. It will be known as nucleotide. But here, it's not mentioned about phosphate group. It's not mentioned about phosphate group. So, can I call it nucleotide? No, I will just call it nucleoside. Okay, side. Tide comes, okay, tide is like this, right? So similarly, when the phosphate comes over here, it will be like tide. So phosphate comes like this, 3 phosphates comes. So it's like tide, they come, but then it breaks down, only 1 phosphate is left. It is known as nucleotide. Okay. Let's move ahead. Okay, question number 3. Removal of RNA polymerase 3 from nucleoplasm will affect the synthesis of what? Very important. We have 3 types of RNA polymerase. 1 alanine polymerase 1, and alanine polymerase 2, and alanine polymerase 3. So what will happen if I remove the RNA polymerase, or what do you call it, 3? 3A. Children, can you answer just 3A, B, C, D? In that way I will remember it better. Okay. Oh my God, so many people out there. You are saying tRNA. You are saying tRNA, are you correct? Yes, you are correct. Always remember this thing, okay? RNA polymerase 1 helps in formation of rRNA, ribosomal RNA, okay? A ribosomal RNA. RNA polymerase 2 will be helping in transcription of hnRNA, and also it will be made to mRNA later on, okay? Now, RNA polymerase 3 will be basically helping in tRNA. There are other forms also. Please remember, go through NCERT. We will be able to, understand better. Okay? Nijaganaradhya, explain every option and their function. This is what I explained. Okay? So, rRNA, ribosomal RNA, the RNA that you need to form the ribosome is over here. rRNA is being coded with the help of RNA polymerase 1. RNA polymerase 2, hnRNA. Okay? And RNA polymerase 3, tRNA. I hope you got it correct. So your answer will be tRNA. Remember this, very important. Okay. Next question. Who first got the X-ray crystallography photographs of DNA? You answer that and I'll tell the names of the children who are there. Monisha, good education can change anything, but a good teacher can change everything, and that's you, ma'am. That's so sweet of you. Thank you so much from the bottom of my heart. Muslim, Suvida, Narasimha, Kiki, Nijaganaradhya, Ravi Kumar, Sonia, Saina, Chandrasekhar, Angeline, Sudeep, Devraj, Preeti, Kavita, Angel Gaming, Gagan, Ravi Kumar, and then Pradeep, Kizar, Aditya, Sonia. Oh my God, yes. So many people out there. Nikhil, Sharada. Okay, so my dear children, the first person to basically, or the first set of people who got the X-ray crystallography photographs of DNA was none other than Wilkins and Franklin. Wilkins and Franklin. And you know what? Who used them? Who used them for deducing the DNA double helix model? It was Watson and Crick who basically took this photograph, studied by themselves, and then came out with a double helix model of DNA. So double helix model of DNA was given by Watson and Crick. And who gave the extra photography for that? Wilkins and Franklin. So your answer becomes D. All of you who got D as the correct answer, kudos to you. You're going great. Okay. Now going with question number 5. What happens to DNA in S strains of pneumococcus when it was subjected to heat killing? So this is a very cute story that we have. So there is 2 types of strain of bacteria pneumococcus. One is S strain and one is R strain. This S strain bacteria has a smooth covering over it. So everything that looks good might not be good. Similarly over here, the smooth strain is the one that can kill the mice, my dear children. If the mice is affected by the smooth strain, it will be killed. So basically what was done is that this S strain was heat killed. That means it was treated with heat. It was treated with heat, and when it was treated with heat, it was not able to kill the mice. Why did it happen so? What was happening inside the S strain? Basically, DNA is like this. When you provide heat, the complementary strands, they will be separating out. They will what? Separate out. So that is going to happen, which is why it is not going to be functional. Okay, it is not able to kill the mice. So what happens to the DNA in S strain of pneumococcus when it was subjected to heat killing? Phosphodiester bond was broken? No. 2 complementary strands were separated? Yes. I hope you have studied polymerase chain reaction. Okay, in that first step is denaturation, where what you are doing, you are providing heat so that they get separated. Similarly over here also same funda is being utilized. Not to amplify the number, but yeah. So your answer becomes B. Okay, so those of you got B, and those of you who understood this, do not forget to like and put hearts. Okay, if you are not understanding, ma'am, why it's not D? Broken into fragments. It can be broken into fragments, but for breaking into fragments, you need to break the bonds out there, and that is not happening. and that is not happening in the case of what's the S strain. Remember, if you remember, my dear children, the S strain, when the heat killed S strain was mixed with R strain. The R strain was transforming into S strain. Why? Because over here the complementary strands separated, but over here when you kept them back, my dear children, in this condition, they were going and binding to the strands over there, forming an S strain back again. Forming an S strain back again. So if it is fragmented form, you cannot revert it back. So there is no fragmentation happening in this particular heat condition. When you provide heat, it is not going to get fragmented. For that you need enzymes. For that you would be needing enzymes to make the DNA into fragments. Okay, here you are not giving any enzymes, just heat. In heat condition it will just denaturate. Okay? Okay. Next question, 6th. Transfer of coded information takes place from DNA to mRNA prior to protein synthesis. This is known as. Nikhil, I hope you understood it now, Baba. Okay, and Angeline, why not? It's D. I told you. I hope you got it. Now all the other children who are answering, oh my God. Great, kudos to you. So here the question is, transfer of coded information from what? From the DNA to the mRNA prior to protein synthesis is known as, what do you call this process as? This process is known as transcription. This is known as transcription. And now from mRNA, when you are making a protein, what do you call this as? Translation. So always remember this in this format. Whatever DNA speaks, protein does not understand it. So that is why you need an mRNA to translate all the information. It is like message. It is the messenger RNA. That is why mRNA, it translates all the information. So here you have translation, here it is transcription. So B becomes your answer. Okay? Oh my God. Someone says B. Okay. Now question number 7. In which of the following organisms RNA acts as a genetic material? Tell me. Now you tell me. Now let me see who all are fast. Okay. Angeline says D. 6B. Chandrasekhar says B. Devraj B. Guruswamy B for the previous question I am saying. Okay. Nijganaradhya, Preeti, Devraj, Angeline, Devraj, Varsha, Basweshra, Suvidha, Nikhil, Nikita, Manish, Angeline, Spurti, Sumit, ma'am, we love you. I also love you. Okay. And Varsha, Devraj, Nikhil. Okay. Children. Okay. J is back over here. Bindu Shri, oh my dear. Okay. D. So yes, children who got a D, you are absolutely correct. Okay. So TMV is tobacco mosaic virus. Okay. Q beta bacteriophage. Both of them basically would be having RNA as the genetic material, my dear children. East, no, does not have RNA as genetic material. It will be having DNA as a genetic material. So basically, your answer becomes both A and B. So this is D. This is little bit outside NCERT, not completely, but yes. So TMV, tobacco mosaic virus, and Q beta the bacteriophage, both of them are going to be having RNA as genetic material. Okay. Now going ahead. Question number 8. Lac operon concept was proposed by. Now let me see who will remember those names. And let me read out the other names. Chandrasekhar, Suvidha, Sharadha, Bindushri, Devaraj, Spurti, Angeline, Sonia, Varsha. I see the girl power out there. Okay, come on boys also. Come on girls and boys. Everyone out there, give your best. 8A, I see so much. Yes. It is A. Lac operon concept was proposed by Jacob and Monod. Please remember, very important thing. Okay, Michelson and Stahl experiment, what was it for? It was for DNA, semi-conservative replication of DNA. And other things, you don't have to remember. Nirenberg was the one who spoke about genetic code. This and this, not needed. But at least these things you should be knowing. Okay, Jacob and Monod, basically lac operon. I hope you got it correct. Yes. Question number 9. Human Genome Project was not started in which year? Now that is again an intricate detail. Okay, Kartikeya Varma, sorry for the intrusion ma'am, but I had a few doubts regarding the crash course, but no one picking up the call at the given helpline number. Can you give your details in the comment section? We will reach out to you. Okay, not exactly, don't give your phone number and all. Okay, if you can at least tell the name, or probably you can put this. Okay, ask edutube @Deeksha learning.com. Can you just mail here If you can mail here, it will be really good. Ask EduTube, it's T. Kartikeya, can you do that? I hope that will help you. Yes. It was ended on 2000. I am asking about when was it started in. So lots of people are saying B, your answer is B, it is 1990. Human Genome Project was started in the year 1990, please remember it. Okay, going ahead. Okay, now find the mismatch. Again, something that I need your answer from. I need you to answer. Okay, okay. You have theta bacteria, 174 bacteriophage having 5386 nucleotides, bacteriophage lambda as 48,502 base pairs, E. coli with 4.6 into 10 to the power 6 base pairs, haploid human genome, human DNA 6.6 into 10 to the power 9 base pairs. What is the mismatch over here? Oh my god, you guys are challenging. Okay, Chandrasekhar, Guruswami, Varsha, Spurti and Varsha Devraj again, Mallikarjun, oh, new people. Okay, all of you, Kavya, Angeline, Spurti, Nikhil, Kiki, Syed, okay, Syed, every one of you are correct. This is wrong, my dear children. Why? Haploid human DNA, that means half the amount of human DNA is found in what? In the gametes, my dear children, in the gametes, and it will be 3.3, not 6.6, that is totally wrong. Okay, it will be 3.3, remember that. And one very important thing, where do you find it? Okay, tell me one thing. Do you find the haploid content in primary oocyte? Tell me in answer yes or no. Your question is, do you find haploid content of DNA in primary oocyte, yes or no? Yes or no? No. Priyadarshini, no. Okay, Veer, okay. Ma'am, can I get good rank if I start studying KCET from now? Yes, please start studying. You will be getting some good rank if you start studying 100%, say 110% efforts if you're going to put. I am pretty sure you're going to get better. I am not obviously going to joke around here saying that you might get great ranks. I do not know about that because depends on your preparation, but definitely you should give it a try. Okay? So, no ma'am, no ma'am, no ma'am. Yes. So, basically in primary oocyte, my dear children, it has just started off the division. It is still in meiosis 1. Okay. It has not become haploid. It is diploid. So, it won't be happening. In secondary oocyte, it will be haploid. So, please be careful about that question as well. So, this is the mismatch that we are having. Your answer is D. Okay. Now, going ahead, question number 11. Which of the following radioisotopes is not suitable for DNA labeling based studies? For example, I want to label the DNA. Which of them will be not suitable for DNA labeling? Veer, you are most welcome, Baba. Okay. Yashoda, Babu Chavan, so many new names out there. Okay. Kavya, Sudeep, Devaraj, Syed, Spurti, Angeline, everyone, where are you? Nikita, Varsha, Guruswami, where is Jai? Where is our Sia Shetty? All of you come back. Okay, Jai is here. Okay. So when I call Jai, he is back. Or he or she, I do not know who is that. Satish, you are not understanding. Baba, what are you not understanding? Tell me. If you are not understanding the question, I can repeat you. Basically, I want to label the DNA, radioisotope. I mean, I want to make this DNA show up in under autoradiography machine. So for that, I have to label it. I have to use certain things to make up this DNA which are radio labeled. So what are the things that I am not going to use? That is the question. What are the things that I am not going to use? Kiki, you are here. Thank you, Baba. Okay, that is the question. So which of the following is not suitable for DNA labeling? You are saying, everyone is saying A. Devraj said D. Devraj said D. Everyone else is saying A. My dear children, listen to me carefully. In DNA, you have the pentose sugar. You have nitrogen base, okay, you have phosphate group, agreed? This is carbon, this is made up of carbon. Carbon has hydrogen also, okay, around it. Obviously, carbon will be bonded to hydrogen also. So, I have to label everything, I can label everything that's up over here. Okay, nitrogen base obviously will be having nitrogen, so I can basically label the nitrogen. I can label hydrogen also because it is a part of this. I can label phosphate also, but is sulfur a part of it? Is sulfur a part of our nucleotide that makes up the DNA? The answer is no. Sulfur is not the part of the nucleotide that makes up the DNA. So basically even if I use this, it won't be radio labeled because sulfur is not at all taken. Ma'am, It does not contain sulfur. No. In proteins and all you might have it. In this you do not have sulfur. Always remember. Okay. So your answer becomes D. S35. Remember the experiment done by this guys. What is the name of that experiment, children? What is the name of that experiment? Come on, come on, come on. Hershey and Chase experiment on bacteriophage, where he was labeling the protein coat with S35, but DNA was coated with P32. If you remember, why DNA was coated with P32? Because DNA is made up of phosphates. Okay. So your answer becomes D. I hope it's clear to you. Everyone who got the answer clear, give me a thumbs up or give me a heart. Give me a thumbs up or give me a heart. Fast, fast, fast. Now I get it. Yes. Measles and Stahl experiment, it is N15, N14. That is different. Yeah, in that also you're taking N, no? So yes. Great, great, great. Okay, you got it. Thank you so much. Sharda, Priyadarshini, Babu Chavan, everyone out there. Now 12th, Devraj. Thank you, thank you, thank you. Question number 12. Euchromatin differs from heterochromatin because euchromatin is what? Tell me the answer for this. What is euchromatin? Basically, our DNA is very long. You want to make it into compact nature so that it can be fitting inside the nucleus. So what do? You have certain histone proteins. You have certain histone proteins. How many of them? 8 of them. 1, 2, 3, 4, and then 5, 6, 7, 8. At the back side it is there. Okay. Now on this histone protein, I am going to wrap around with the DNA. 2 wraps. Okay. Twice it will be wrapping around, and this structure is known as a nucleosome. Similarly, you have lots of structures like this. Okay, you have the whole structure like this. You will call this structure as chromatin now. This condensed DNA is known as chromatin. Now, my dear children, there are chances where between 2 histones, okay, there could be lots of space. In this case, I will call this as euchromatin. I will call this as euchromatin. And in euchromatin, they can be stained. They are stained but lightly stained. They are not darkly stained, they will be lightly stained. And over here, since it is DNA, it's open DNA, RNA polymerase can come attach over here and do the process of transcription also. So it is transcriptionally very much active. Okay, and it is loosely packed, as you can see. On the other hand, if I talk about heterochromatin, what is happening? It is very close to each other. They are very close to each other, so they are darkly stained. They are not loosely packed, they are tightly packed, and they do not have space for transcription to happen. So here, euchromatin differs from heterochromatin because euchromatin is loosely packed. Yes, ma'am. Lightly stained. Yes, ma'am. Transcriptionally more active. Yes, ma'am. So your answer becomes D. All of this. Got it? Got it? Yes. Cool. All of you said D. Great, 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 children. Now question number 13. The unusual nucleotide added to the 5' end of hnRNA during capping is known as what? Guru Swami, thank you for responding, and all of you who are responding, kudos to you. Okay. Tell me. The unusual nucleotide added to the 5' end of hnRNA. What do you mean by hnRNA? So basically, you have the DNA and then you do transcription. In transcription, you do not make mRNA directly. You initially have hnRNA. What is an hnRNA? Okay, so basically over here, you will have 2 things, exons, introns and everything. That means region that codes for proteins, region that does not code for proteins. Same like that only, while transcription also, here the RNA will also have this exons, introns and lot of things. Exon, intron and lot of things. So this is not a perfect thing that I can send outside for making protein. I have to make it into messenger RNA, mature form of RNA. So what will I do? I will do so many things. First thing that I'll do is splicing. I'll remove the introns. I don't want them. They do not code for anything. And I'll join the exons. So one is splicing. And apart from that, over here, capping is being asked. What happens in capping? At the 5' end of the hnRNA, my dear children, what has happened? Methyl guanosine triphosphate, MGPPP. Okay, methyl guanosine triphosphate is being added over here. Okay, and then there is tailing also. Capping, splicing and tailing. Tailing where you are adding adenine group over here. You are going to add adenine group over here. And now when it is ready, it is known as messenger RNA or mRNA, and it will be sent outside. Why do you put a cap? Okay, why do you put a cap on your head? So that you remain safe from all the sun and things like that. Similarly, this mRNA has to be safe when it goes outside. Until now it was in the nucleus. It will now go outside in the cytoplasm. There are so many enzymes to cut and cleave it. So to save themselves, you have methyl guanosine triphosphate. Methyl guanosine triphosphate, that is capping. I hope you got the answer. Okay, so all of you who said B as the correct answer, it is Awesome work. If you didn't get it, it's totally fine as well. Okay. Now, question number 14. There are no anticodons in tRNA for the following codons except. Okay, so question, listen to me. There are no anticodons in tRNA for following codons except. Can you tell me? Fourteen D. Lunch, ma'am, would be having lunch really soon. Okay, just after solving few more questions, we'll be taking lunch. That's so kind of you for asking. I hope you had it, or you might be having it. Children, please have your food and just listen. You just have to listen to me, yeah? Ma'am, please take breaks. We will be online, sitting, listening and texting, but you are explaining continuous. Oh, that's so sweet. So sweet of you all. Now, D, Devraj, Suvidha, Charlotte. Okay, you are all saying D. Why, my dear children? Because all the others are stop codons. U, A, U, Ag, U, Ga. Okay, 3 of them are stop codons, so there are obviously no anticodons in the tRNA for that. Okay, except one guy, that is AUC, it will be having it. So your answer is D. Okay, your answer is D. Awesome, children. Question number 15. Rahul also said D. Great, great. Question number 15. In lac operon, in the absence of inducer, that is lactose, repressor protein binds to what? Can you tell me the answer for this, and I'll read out the names for a lot of other people who are not there. Okay, I didn't read out the names for so long. Spurti, Guruswamy, Chandrasekhar, Angeline, Basveshwaraya, and then I had Sania. Hi Sania, I didn't tell your name, I'm so sorry. Nikita, Kiki, Preeti, Nikhil, Jai, and Spurti, Babu, and Ayan, Sania, great, Rahul, Sonia, Varsha, so many names. Yes, won't get 14. Okay, you didn't understand 14th one. So here it is asked, there are no anticodons in the tRNA for the following codons. No anticodons in the tRNA, as in, for example, if I have UAA and UAG, this is the codon. Now tRNA usually have an anticodon opposite to this. For example, I have AAA. It will be having UUU as an anticodon. But certain tRNA, they do not have anticodons for this. Why is that so? Because they are stop codons. They are what? Stop codons. They do not code for any amino acid. And tRNA is the one that carries the amino acid. So if they are not coding for any amino acid, there is no need for anticodon as well. Okay. So these guys are the ones with no anticodons. But over here, it is written except one, all of them do not have anticodons. So it is only one that has anticodon, that is AUC. So if it is AUC over here, my dear children, it will be what? UAG over here. That will be the anticodon. Okay. Yes. Now tell me. In lac operon, yes, we were here. I'm so sorry. Okay. Lac 1. Okay. In lac operon, in the absence of inducer, repressor protein binds to lac 1 I gene, lac P gene, lac O gene, lac ZYA gene. Your answer becomes what? Your answer becomes what? So basically in lac operon, I am just going to tell you. So there is a promoter region, okay, and then there is a repressor protein that is known as basically I gene, regulatory gene, okay, and then there is again a promoter gene, then there is an operator gene, then there is ZYA gene. So this whole thing is one gene, this whole thing is another gene that we are having over here. So usually this repressor I gene codes for repressor protein, and this repressor protein goes and binds on the O. It goes and binds on the O. Why does it go and bind on the O? So that the RNA polymerase which binds on the promoter cannot move forward and it won't transcribe ZYA. That is what it is doing. It is repressing the gene. Okay. But when inducer comes, that is lactose comes. Okay. There is a what do you call it in movies and all. In advertisement, man mein laddu phoota. Similarly, when lactose comes, repressor protein is like, I won't go to operator, I'll just stay here. So basically it is free and now transcription can easily happen. That is the thing. So usually when inducer is not there, lactose is not there, it will go bind on the operator gene. So C becomes your answer. Okay, so C becomes your answer. Got it? Can we move ahead? Devraj, yes, you all are correct. Sania, Sonia, Baswaraj, Jay, students, Purti. Okay, great. Question number 16th. Can we try it out? Children, if you go down, if your comments go down, probably my motivation also would be going down. So keep on answering, okay? One of the similarities between DNA and RNA is that both have similar pyrimidine bases, have similar pentose sugar, are polymers of nucleotides, are always double stranded. What will be your answer for this? RNA and DNA, my dear children, do they have similar pyrimidine bases? We have pyrimidines, two type of pyrimid. You have T and C as pyrimidines in the case of DNA, but in the case of RNA, it is not T but it is U and C. So are the pyrimidines same? Wrong. This statement is wrong. B, have similar pentose sugar. Array, in the case of DNA, you have deoxyribose. That means over here there is just H, there is no O on the second carbon. But in the case of RNA, there is ribose sugar where on the second carbon you have OH group. Okay, that makes the whole difference. So this is also wrong. This is ribose, this is deoxyribose. Wrong. Are polymers of nucleotides? Is it so? Yes. So basically, the nitrogen base come over here, nitrogen base come over here also. There is pentose group here, there is pentose group over here also. So both of them, they are nucleotides and they form stack of nucleotides over each other, and that is how they form. So they are polymers of nucleotides. Yes, that is correct. Are always double stranded? No, RNA can be single stranded. Okay, RNA can be single stranded, so this is wrong. So C becomes your answer, and I see so many of you have answered. That's so great of you. I'll read out your names. Until then, you try to solve this question. Okay, so Devraj, Guruswami, Spurti, Ravi Kumar, Preeti, Babu, Priyadarshini, Sonia, Rahul, Devraj, Sania, JEE student, and Syed, Guruswami, all of you doing great, awesome work. So we are towards the end, so be tight over there. We have to solve all these questions and go, okay? Okay, Muslim also, great. Charlotte, great. mRNA contains 1002 nucleotides, that is including initiator codon and terminator codon, in its reading frame. How many amino acids would be there in a protein synthesized due to this gene? Can you tell me? So how many nucleotides are there, my dear children? 1002, including the initiator codon and the terminator codon. Listen, you have to find the number of proteins that are going to be, sorry, number of amino acids. Okay, so what is your answer? Someone says B, Devraj says B, Sharada, your name was Rekha, right? Okay, anyways, you are saying B. Let's see who are saying B. Priyadarshini also says B, Ravi Kumar says B. So my dear children, in 1002, one is initiator codon. So yes, initiator codon codes for amino acids also, so that's not a problem. But terminator codon does not code for amino acid. And terminator codon includes what? 3 nucleotides. 1, 2, 3. So we have to remove 3 of them from here because they are not going to code for any amino acid. So 1002 minus 3 is 999. You have 999 base pairs now. You have 999 base pairs now. So basically, how are amino acid coded? 3 comes together, and that's how you get one amino acid. So 999 divided by 3 is what? 333. So if 333 codons are there, then 333 amino acids will also be there, which is why your answer is B. And all of you got it correct. Good dose to you. Great work. Okay, great work. Always remember, please remove this and then only try to find it out. Fine. Now, question number 18. Which of the following is transcribed and then translated to form a protein product? Important. Important. Which of the following is transcribed first and then translated to a protein product? Gene for tRNA, intron, exon, leader and trailer factor. Oh my god. Once again explain. Okay, I can explain once again. Until then, please children, you solve this question. So I am taking that question over here so that anyone who is reading this question, they can continue. So we have these many nucleotides. Agreed? Okay. Now, basically what is happening? One, you have a terminator codon. Terminator codon consists of 3 nucleotides. So we have to remove 3 nucleotides because they are not going to code for any amino acids. So I do minus 3, I get 999. Now we have 999 nucleotides. This 999 nucleotides, basically they make a bunch of 3 codons. Okay, bunch of how many codons? 3 codons. So if I want to find how many codons are there, what do? I divide by 3. So I get 333. So I have 333 codons, and each codon, my dear children, codes for one amino acid. So if 333 codons are there, how many amino acids will be there? It will be 333 itself. So I hope you got it, Sanya, now. Okay, now, so 18C, Chandrasekhar says 18C, Rahul says C, and then, come on, Charlotte says B. Sanya, you're most welcome, dear. Guruswami says A. I have so many. Children, your question is, what is that something that is getting transcribed also and then translated also? So to getting translated, obviously it should be there in the mRNA. So we studied in the DNA we had both the introns as well as exons. In the hnRNA, again we have introns, that does not code for anything, and the exons, right? But when we make mRNA, aren't we going to remove the introns? Yes. Now we just have the exons. So exons are the one that is going to be translated to what? Proteins. So gene for tRNA, no. Introns, no. Exon, yes. Leader and trailer factor, that is not even there in your NCERT. Why do you care? Okay. So basically it will be exons. Exons are the one that is appearing in mature or processed mRNA exactly. Okay. So all of you who didn't understand it, I hope you understood it now and you got it correct. Cool. If you understood, can you give me a heart or thumbs up? Children, if you understood it, can you give me a heart or thumbs up? Or if you didn't understand, you can tell me right away as well. Come on, come on, come on. Yes, I see certain hearts flying. Okay, thank you so much. Thank you, thank you, thank you. Okay. Now, during Michelson and Stahl experiment, heavy DNA was distinguished from normal DNA by centrifugation in what medium? So heavy DNA was there. So the DNA that was taking up the N15 isotope, that was heavy. Okay, N15 isotope is the heavy one. Okay, and the DNA that was taking N14, that was comparatively lighter. But anyways, this heavy and light, everything, you have to centrifuge, right? You have to put it in a tube, you have to put it in a media and then rotate it. That's when it gets separated into heavy and light and things like that. In which medium are you doing it? That is the question. So I see Rahul, Ravi and Ravi Kumar, Rahul, Ravi Kumar, Sanya, Rahul again, okay, Devraj, Srishti, hi, Priyadarshini, Aditya, hi, someone new out there, Mahati, Ramesh, so many new people, Hasibu, nice, Aishwarya, Rajappa, Chandrasekhar, Preeti, great going my teacher. All of you are saying B and B is the correct answer. It is cesium chloride, CSCl, JEE student. Exactly. So your answer is B. Okay. Not C, Baba. It cannot be C. NH4Cl is the thing that I am using. Okay. It's the medium where I am particularly growing. Okay. this thing, no. This is the medium where I am growing the DNA. But for centrifugation, I am going to put it in a medium to centrifuge it. And that is cesium chloride gradient. I hope you got it. So your answer is B, not C. Okay. So many hearts, 100 and everything is flying. That's so great. Children, did you like the video? Did you put like on the video? Definitely do that and subscribe to your channel and tell it to your friends as well. Okay, and until what time we will be studying, we will be finishing it right off. Okay, few questions are there. 20th question, do it. Which of the following is not correct with regard to outcome of HGP, Human Genome Project? I see 41 people out there. Kudos. So what is not correct to outcome of HGP? Human genome consists of 3,164.7 million nucleotide bases? Yes. Chromosome 1 contains 2968 genes? Yes. Less than 2% of genome codes for proteins? Yes. There are about 5.5 million locations of single nucleotide polymorphism. What will be your answer? It will be D. So what is the correct number? Can you tell me? What is the correct number? What is the correct number? If it is not 5.5 million, what is the correct number? Anyone out there? So let me see who are getting the answers correct. Hasibu and then Chandrasekhar, Devraj, Priyadarshini, Shrishti, Aditya, Sanya, Babu, Hasibu, oh my god, JEE student, Kiki, Preeti, Rahul, Ambika, hi. Okay. 1.4 or 2.4? Can you confirm? It's 2.4, right? Open your NCERT. It's 2.4 or is it 1.4? Check. That is for you. That is for you to tell me the answer. I am not going to share it over here. Open your NCERT. Go in the HGP page. Okay, towards the end, the last point, it will have the answer. At least that I have told you. Okay. Oh, have you finished the questions? Yes, we have. Okay, there are 60. Okay, there's 20 questions and I didn't know that we are gonna finish it, but yes, now I am ready to take up your doubts and thank you for being here live with me and keep on telling that. And ma'am, it's 1.4 million, look, pakka. So I'm locking it. Should I lock it? It's 1.4. My dear children, if you have any doubts, do tell me, and right now, right away, like the video, comment down whatever you feel of this class, and also if you want PDF will be given to you in the channel. If you tend to forget, please put it down as well. If you have doubts, you can ask me right away, or you can put it down in the comment section. Now I remember someone had a doubt on law of segregation. Someone had a doubt on law on segregation. I can explain it doubt to you. I don't remember who asked me. Kiki says, I literally love your bio videos. Thank you so much. That means a lot. And Preeti, thank you so much. Sharda, thank you so much. Children, ask your doubts or we shall end the live. Rahul, thank you. Aishwarya, oh my God, I see so many. Yes, you asked. Sharda, I hope you were asking about law of segregation, right? So law of segregation, basically it states that, suppose I have a parent number 1 having capital T, capital T, small t, okay, and here also capital T, small t. This is parent number 2. So law of segregation says that when you are forming the gametes, the chromosomes which are there, they are going to separate, and only one chromosome are going to come in the gamete. That means either the gamete can contain capital T or the gamete can contain small t. Both of them would not be coming together. This is law of segregation. That is why we have one chromosome per gamete. For each character it will be like that. In 23 pair of chromosome, only one chromosome is going to come from both the parents. Okay? Yes. Thank you so much. Oh my God, I feel so happy. Ma'am, when is your next class? We would be having it on Monday, Tuesday, Wednesday, Thursday. Thursday will be my next class. Okay, and children, you can keep on asking me doubts as it's there. Do you have any doubts or shall we end the class? Sharda, did I answer your doubt, Baba? Is it clear? Let me read it out. So Rahul says, thank you, you're my favorite. That means a lot. Shrishti says, thank you. Suvida says, thank you. Thank you so much. Kiki, if JJ ma'am is on live, oh my God, I'll be there. Aishwarya, thank you so much. And nice, I like your classes. Thank you so much. Sharda, okay. So all of you have got it cleared. Any other doubt from molecular basis of inheritance or principles of inheritance and variation? I'm standing for that. If you have anything, you can ask me, or shall we end it? Either way. Aisha, thank you so much for your efforts. Thank you for you too. Okay. No. Can I get 7? Mumtaz is asking 700 in NEET for the first attempt. Yes, you can, if you give your best. There are so many things associated with it. And children who are there over here, NEET channel is there. Okay, subscribe to the NEET channel if you're someone who is going ahead with NEET. There I'll be guiding you, and all the teachers will also be guiding you. So please be there if you are someone who is out there. The name is Deeksha NEET Campus. There are free mock tests for NEET that you have to definitely take. So in that channel I have given you the link for the free mock test. Make utilization of it. It's free, totally free. You just have to register and just attempt. Okay, so we are going to take break, and yes, I wanted to say one more thing. If you are someone, yes, KCET students, my dear children, there is Abhyas. If you know, there is Abhyas KCET test series. Okay, where you have full length and part test, mock, test over there. You have to obviously pay it. The rupees is rupees 1498, which is not very high, my dear children, because in here you are getting so many things. You get the paper, part test and full length test, including both PU1 and PU2, and apart from that video solutions are also given to you. So please make utilization of that. You have to you can find it in the description box. Okay, if you're not finding it, do let me know. Abhyas KCET test series, take it. Okay, so that's all children. Do not forget, go check it out once. If it is useful for you, you should definitely buy it. Yeah. So already subscribed. Thank you so much, children. So I'll go, and lots of love to all of you Chutku people. Wishing you all the best. You are going to ace the examination, and go watch all the top 30 videos. Very important. I've tried to explain my level best in all those things. So bye bye. Signing off. Aye Captain. Okay. Take care. Love you a lot. Bye bye. Will you also make one shot for NEET? One shot for NEET might not be coming around, but we have already, okay, so PYPs will be coming, okay, previous year papers. So past previous year papers, and also chapter wise PYQs also will be given, the high weighted chapters that would be given to you, because you have so less time, right? So this point of time, delivering the best thing would be more useful for you, I think. Yeah. So that's all. Bye bye. Take care. Lots of love and wishing you all the best. Bye bye.